Sinai School of Medicine. Thank the organizer for the great opportunity, and I'm very glad to present our recent work on single molecule categorization of bacterial epigenomes. I mainly covered two of our recent work on this topic. The first one is on the epigenetic complexity and heterogeneity in pathogenic bacterial, which is done by John Bellario, PhD students in the lab who's in the audience. And the second one is on the functions that are epigenetically regulated in pathogenic bacterial, done by Shijia Zhu in the lab. And towards the very end of the talk, I will cover briefly about the recent discovery of novel epigenetic marks in mammalian genome in collaboration with Andrew Shell at Yale University. So it is very well known that the information content of DNA is not only carried by the DNA sequences, but also by the chemical modification to the DNAs, such as DNA methylations. However, very importantly, when we talk about DNA methylation, most of the time we specifically refer to a specific form of DNA methylation called 5-MC. Here the 5 means that the methyl group is added to the 5th position of the cytosine base, right? 5-MC has been extensively studied in many eukaryotic species. And for example, 5-MC play a very important role in the healthy development and also in the gene silencing of vertebrates. And even more excitingly is that for many human diseases, people often observe that there are differential methylation patterns between the normal cells and the disease cells. In this example from a lung cancer study, not only you see the difference between normal and disease cells, you also see that the subtypes of lung cancers have differential methylation with each other. So with all of this rich understanding and information about 5-MC in eukaryotes, you may wonder, okay, for bacterial, let's just go ahead and study 5-MC in bacterial. However, to better study this question and have better understanding, we need to step back a little bit, try to look at the whole universe of the DNA methylation in the whole life. So this is the universe of chemical DNA modifications. This is 5-MC. I use a very small font so you can get a sense that there must be many other things. Indeed, 5-MC is only one specific type of many other methylation or modification to the cytosine base, right? There can be 4-MC, which means the methyl group can be added to the 4th position of the cytosine base. And 5-HMC is another derivative of 5-MC, right? And also, if you go beyond the cytosines, methylation can also happen to the adenine base, which is, for example, 6-MA, right? And even beyond that, if we talk about some other types of DNA modification beyond methylation, there can be a large number of DNA damages that can happen to DNA, either caused by oxidation or UV light or smoking. And some of these DNA damages can lead to basically mutagenic, so that can lead to some diseases. But today, our focus will be on the bacterial methylome, which is comprised of these 3 types of DNA methylations, 6-MA, 4-MC, and 5-MC. If we take a closer look at these 3 types of DNA methylations here, you can see 5-MC in bacterial actually only represent 10% of the DNA methylation landscape, which means if we really want to study DNA methylation in bacteria, we have to look at the other, especially 6-MA, which is represent 80% of the landscape of bacterial methylome. So, but you may ask a question, so why do we want to study them? So what are the functions of DNA methylation in bacterial? In fact, DNA methylation was first ever discovered from bacterial, which is in the context of a restriction modification system. And there has been a really rich study more than tens of years. The people identify that DNA methylation play important roles, including regulation of cell cycle and DNA replication and mismatch repair in bacterial. But what I will focus on today is that recently more and more evidence is suggesting that DNA methylation also have a very general role to regulate gene expression, virulence of pathogens, and also the antibiotic resistance. Many of you may heard the recent shocking news that in some superbugs are discovered in the beach close to Rio Olympics. And by superbugs, they basically mean those bacterial are pathogenic, and also they're resistant to a large number of antibiotics. So this clearly shows that the study of the basic science of bacterial methylome clearly have a connection to the public health. So now if we want to study DNA methylation in bacterial, and how do we detect them? And it's well known that bisulfite sequencing can detect DNA methylation, but specifically of 5-MC. But for the other 2 types of DNA methylations, actually until recently, no technique can detect it at genome-wide scale and at very high resolution. So the game changer in about 4 years ago is third-generation sequencing, which is represented by the Particular Biosciences, or you may know the PacBio, or Oxford Nanopore, and the third one you may not hear very often, but there's another one called Genia. And one of the most commercialized technique, third-generation sequencing technique is called smart sequencing, single molecule real-time sequencing. In smart sequencing, each DNA molecule is monitored by a DNA polymerase at real time through the DNA synthesis. So you can see that during the synthesis, these fluorescents are marked, are used to mark the individual nucleotides, so you can see what each of these individual bases are. But the real magic of this real-time sequencing is that not only does the DNA polymerase capture what bases you see, but also the time it takes for the DNA polymerase to translocate from one base to the next one. This unique time dimension, which we call interpulse duration, is actually highly associated with the DNA methylation status. And in this example, you can see two DNA templates here. They have exactly the same DNA sequence. The only difference is that at this base, the A is methylated, 6-MA. This one is not methylated. You can see it takes much longer for the DNA polymerase to translocate from one base to the next one. And if you can imagine, we have a native DNA and we have a kind of methylation-free DNA. If we take a ratio of the IPDs, then we'll be able to detect where the DNA methylation are. In this example, you have two 6-MAs. You can see two primary peaks on top of the 6-MA. You can also see there are two secondary peaks right after. So after we realized that it's really exciting that it's possible to map a 6-MA at genome-wide scale at a single nucleotide resolution, it was back in 2011 when there's a kind of well-known E. coli outbreak in Germany and with a lot of high increased virulence. So there's many groups across the world trying to ask, so what gene contribute to the virulence of this outbreak strain? But we try to ask a different question. We want to say, beyond the DNA sequences, is there any methylation that further contribute to the virulence of this outbreak strain? So we use smart sequencing, we map the first 6-MA methylome, and indeed we find that this outbreak strain have a unique methylation pattern. So basically there's a newly acquired element through a horizontal transfer. This element not only include additional virulence genes, but it also include a methyltransferase. This methyltransferase, if you imagine this just one single gene, but it methylate more than 2,500 adenine sites all over the genome. So we immediately ask whether this have an impact on the gene expression of bacterial. So through perturbation experiments, we knock out an overexpression. What we notice that more than 1,000 genes is differentially expressed when we change the expression of the methyltransferase. And that represents more than 20% of the total genes encoded in the E. coli genome. So these three pathways are enriched for both methylation and the gene expression. So this is the first study, but right after that, in the past 4 years, there are more than 700 bacterial methylomes have been mapped. So nowadays, I can say is fairly reasonable that people and fairly accurate people can detect methylation motifs for every single bacterial that you can extract the DNA from. And also you can detect the methyltransferase from the genome, and you can link the methylation motif to the methyltransferase. However, if we only represent this bacterial methylome in this way, which is the current standard, the motifs do not capture the dynamics of the methylation status at different time points. For example, if you treat the bacteria with antibiotics. Also, it does not capture the cell-to-cell heterogeneity within the bacterial population. So why do we care about the cell-to-cell heterogeneity in bacteria? It is because the context is bacterial, although we assume it is very clonal, very simple, actually it's very smart because they just try to create a lot of diversity at the phenotype level. So whatever drugs you use, or maybe through the course of infection, they just randomly create a lot of these heterogeneity and very hard to battle with them. And one way that bacteria can create a lot of these heterogeneity is through the kind of heterogeneity at the molecular level, at the gene expression level. And further upstream, you can see that epigenetic regulation can indeed contribute to the gene expression levels. So if you look at all of this together, is that although in a very clonal bacterial population, a lot of heterogeneity will happen at the epigenetic level, at the gene expression level, and then eventually at the phenotype level, that give the whole population of the bacteria higher advantage to survive different kind of conditions. However, there's until kind of basically the problem is there's a new method that can really detect all of these heterogeneity. So that's the motivation of the first work that I will present. A little background is that in smart sequencing, each double-strand DNA is sequenced by the DNA polymerase. And there are two adapters on two ends, so you can actually sequence the first strand and second strand again and again and again. So this is the way that we can generate multiple subrays, what we call for the positive strand and also negative strand. So although the technique, it can capture individual molecule, the IPD information, but before this work, all the pre-works use the aggregate analysis because it's very challenging when you really do single molecule analysis at the real time, a lot of errors, different kind of errors. So that's why a previous analysis used this aggregated detection. But we designed a novel method and framework that we can not only detect these methylation and a single molecule and a single nucleotide resolution, but we can also face them using a statistical algorithm that face them at the single molecule resolution for better understanding. So we applied this novel method to a bacterial called Helicobacter pylori. This is the pathogen that causes a large fraction of the gastric cancer every year, so worldwide, and 50% of the world population colonized with this bacterial. So, and as you can see that after we apply this method to this bacterial, although this is very clonal bacterial, you can see we will be able to very sensitive detect there's some subpopulation that have methylation turned on, and in the other cases we have a subpopulation that have methylation turned off. And when we isolate the group kind of and compare them in terms of gene expression, we see they have very distinct gene expression patterns. And so now if we imagine that when we have a change of condition, maybe antibiotic treatment, then some of the subpopulations will have higher fitness, so will be selected for, and then there will be kind of swap over of these different subpopulations in these different antibiotic treatment. And this is exactly what has been observed in the recent study on the H influenza. The agent caused a lot of the childhood ear infection, and also in our recent study on Vibrio cholera, which is regulate the cell envelope stress response. And there's some other bacteria also. So, and let me just use one slide to talk about what's the mechanism of IgG regulation in bacterial. So you may wonder, is it like in eukaryotes mostly repressive, or it can be or is it activating role? The answer is it can be both, because it really depends on the competition between the DNA methyltransferase that methylate the DNA with other DNA binding proteins. For in this example, it can be a TF. If TF bind the DNA first, then the methyltransferase cannot access the DNA, so the gene will be turned on. And if the methyltransferase access the DNA first, the methylated DNA, then TF cannot bind here. So now, this methylation will be repressing the gene expression. But if you imagine this TF is not a TF but an inhibitor, then the same methylation can actually inhibit the gene expression. And there's another kind of, this is a single locus model, but there can also be a global model that multiple genes are regulated by gene expression simultaneously. So now I want to switch gear because I've been talking, so far I've been talking about the bacterial all through, and many of you, I'm sure, will ask about does 6-MA also exist in mammalian genome? There's a really long debate in the past, maybe 40 years. One study 40 years ago discovered 6-MA in mouse, but then almost no study can really reproduce that and confirm that. So there's a really long debate. But very excitingly, that recently through the collaboration with Andrew Shell at Yale Lab, led by his lab, we actually indeed see 6-MA in mouse embryonic stem cell. The reason that why people have been really hard to see it because the level is really, really, really low. It's almost kind of 3 orders of magnitude lower than what you expect from the bacterial cells. So we had to use very sensitive kind of ultra HPLC methods coupled with mass spec. We'll be able to see that out of 1 million adenine bases, you can only see 6 methyl 6-MA, right? This is really rare, but then we also discover 6-MA demethylase. So after we knock out this gene, then we'll see a much increased level of 6-MA. Now the question is, and even you may ask, and even we asked ourselves earlier, that if it's really rare, then is it even functional at all in mammals? The question, yes, it is functional because it's rare but highly specific to certain regions. What we found is that 6-MA in mouse embryonic stem cell is specifically targeted to the young full-length line 1 elements. A little background is the L1 is a retrotransposon that constitute more than 15% of the human genome. So that's really a huge kind of component of the human genome, and it specifically targets the young L1s, but not the old L1s. And when we perturb the 6-MA status across the mouse EI cells, we see not only the differential expression of these L1 elements themselves, but also the nearby genes and also some of the enhancers. So this shows that it have a very specific location and also specific function. And actually beyond these mammals, a recent study also shows XMA exists in a large number of both lower and higher eukaryotes. And so basically it opens a very exciting kind of paradigm to study the mammalian epigenome also beyond the cytosine, the 5-MC methylation. So now here's the summary. I hope I kind of gave you some idea about these 4 major points. First, bacterial methylome have 3 types of demethylations, 6-MA, 4-MC, and 5-MC. Secondly, we developed a novel method for categorizing epigenetic heterogeneity, which is very important to study bacterial, because epigenetic heterogeneity will cause transcriptional heterogeneity and then phenotypic heterogeneity that enable these really superbug and antibiotic resistance to survive our treatment. And eventually 6-MA also exists in mammals and also plays very important roles. So there's clearly a lot of unknowns to be discovered from this kind of new directions. Now John and Shiya are the 2 kind of members in the lab who did most of the work, and also acknowledge the colleagues at Mount Sinai School of Medicine, and our collaborators, Blizzard Lab at NYU, Water Lab at Harvard Medical School, and Shell Lab at the University, and Ho Lab at the University of Chicago, and the funding agency of this. So this is the end of the talk, and thank you for your attention. This is